I will start with this perspective published on the New England Journal of Medicine 3 weeks ago. You know very well that President Barack Obama decided to invest something like $1 billion in the initiative of precision medicine, finally. And the idea is to launch this program. Like JFK decided to go to the moon, he decided to launch this program of precision medicine to bring us closer to curing disease like cancer and diabetes, and to give all of us access to the personalized information we need to keep ourselves and our families healthier. So is this reality? This is the first question. But what is precision medicine, finally? I would like to mention a society that is called Foundation Medicine in the United States. Since 2 weeks now, I know that Genentech invested $1 billion to buy Foundation Medicine, but there are also other societies like Ceres or others in Seattle or in Arizona. And what they do is finally to send to them a tissue sample, and they will sequence all the genome of this cancer, and they will give to you a report like this in which they define the molecular alteration that they find, and they list all the clinical trials that are available for the treatment specifically of that tumor. So there is really something like a commercial interest to sequence all type of tumors, all type of metastatic disease, with the potential aim to identify specific clinical trials that are ongoing to offer to the patient a specific treatment. In the reality, it happens in less than 10% of the cases. So a trial has been completed in breast cancer on precision medicine. It has been published on the Lancet Oncology 1 year ago. The trial was based on metastatic breast cancer patients. They did a biopsy. The biopsy was molecular profiled. And then after identification of specific molecular alteration, let's call them driver pathways, a treatment has been proposed. Just to summarize this trial, the SAPPHIRE-01 trial, the PI was Fabrice André, they screened 404 patients, and just 40 patients received a treatment based on a molecular alteration. No one of these 40 patients received a treatment within a clinical trial. In most of the cases, they received a therapy with an off-label approach. Let's say they found an epidermal growth factor receptor mutation and the patient received elrotinib or gefitinib. They found HER2 amplification in a tumor that previously was not HER2 positive and they received trastuzumab. Or in some cases, they found an amplification of fibroblast growth factor receptor and they received a treatment with lucitanib. So 400 patients, 40 with a specific molecular alteration, and in terms of response, 9% of partial response with a duration of less than 3 months. So this is the impact in the real life of precision medicine. In terms of outcome, 9% response of a duration of less than 3 months. We have to think about this because I will show to you that many institutions in the world are launching programs of molecular screening, and many institutions are creating platforms of phase 1 in order to have different type of approach, but finally the impact on the life of patients is very, very limited. This is a slide of Francesco Bertolini. He presented this 3 or 4 years ago, I remember, following the presentation of Burt Vogelstein on the concept of passengers and drivers. Yes. And finally, what is trying to do precision medicine is to identify, finally, the driver pathways also for gastric cancer. So how do we treat cancers now? Until now, selecting therapy based on tumor histology. In some cases, based on tumor molecular profile. And specifically in gastric cancer, I would like to mention this manuscript. You know that we have the TCGA initiative. We have the Cancer Research UK that did a huge effort in order to sequence all the solid tumors. And this is the genetic and epigenetic landscape of gastric cancer. I did a summary then to identify which are the molecular alterations in gastric cancer. So specifically, we have several types of mutations that are quite similar to the other solid tumors. P53, PI3 kinase alteration, FGFR amplification, AKT, HER2 amplification. This is the summary of the genetic alteration of gastric cancer, derived from the manuscript I showed to you, and from the Cancer Genome website. So these are the potential pathways for which we have a specific drug. So since we have to talk about small molecule against gastric cancer, we have AKT mutation in 1% of gastric cancer, ALK amplification in 3 to 7%. So let's say that crizotinib can be an option for these patients. We have BRAF mutation in 1 to 3% of gastric cancer, epidermal growth factor receptor mutation in 10 to 35, HER2 amplification in 5 to 15, KRAS mutation in 3 to 5, the MEK amplification, despite a lot of trial, according to this genomic landscape just published, is 1%. And then PI3 kinase 5 to 15%, ROS 1%, and RET 1-2%. So very limited, finally, the opportunities to treat a gastric cancer according to the driver alteration for which we have a clinical trial available or a specific small molecule available. So what I can tell to you is that recently, in October 2014, there was an FDA initiative for the design of the clinical trials in the era of genomic medicine. We have two types of clinical trial. The first one is the umbrella design, in which you have one single molecular alteration and then several types of disease. And you will give the drug to the patient that will have that specific alteration. The other type of design is the master protocol. So you start from gastric cancer, you identify, you perform an exome sequencing or a wide genome analysis, and then according to the molecular alteration, you stratify patients with a specific treatment. So it's a very complex initiative to do trials like this. There is one trial ongoing in the world that is the Aurora trial for breast cancer, that is a master protocol in which you perform next generation sequencing in Cambridge. You identify the molecular alteration, and then for any druggable molecular alteration, you have a specific trial. So if Notch amplified, a trial with Notch inhibitor. If PI3 kinase mutated, a trial with PI3 kinases inhibitors, and so on. You need a huge effort, a global effort. In order to identify one patient with a PI3 kinase mutation, you need to screen 100. And think about gastric cancer. It will be the same if you would like to start with the precision medicine initiative in gastric cancer. And don't forget about evolution, genomic evolution, because once you have a progressive disease, for sure you will have another driver pathway. Because if you target HER2 with trastuzumab and then you have a progression to trastuzumab, you will have another driver pathway. This is a very interesting study published from our friends from UK, in which they performed biopsy in different sites of metastatic disease in a kidney cancer patients under treatment with an mTOR inhibitor. And they discovered that some mutations are shared between primary and mets. Some others are completely different. So when you do a precision medicine trial, are you sure that if you don't find the mutation in the liver met, you will find maybe in the lung met or in another site of disease? So many considerations should be done when you approach the patient within a trial of precision medicine. So how to overcome resistance? So maybe, I don't know if it works or not, maybe angiogenesis inhibition can be an example. I am sure another speaker will speak about this, but anyway, since this is a monoclonal antibody, I decided also to mention the recent data on ramucirumab, and I would like to thank you, the patient, my colleague working in the gastrointestinal unit, Dr. Galdit, for providing me this data. This is the recently approved monoclonal antibody in gastric cancer, in patients with metastatic gastric cancer, following the data of the RAINBOW trial, that is a double-blind randomized phase 2 trial. Ramucirumab is a vascular endothelial growth factor receptor 2 antagonist that specifically binds and blocks activation of the receptor. It's more specific than other anti-angiogenic drugs. These were the data of the REGARD trial, ramucirumab monotherapy in second-line gastric and gastroesophageal junction cancer. It was a phase 3 in which the drug was compared to placebo. And in terms of efficacy results, there was a benefit for the anti-angiogenic drug with respect to the placebo. These are all other data of efficacy of ramucirumab that is very similar, finally, when we compare to supportive care or other type of agents. So the median overall survival on monotherapy was around 5.2 months. If we compare to docetaxel as monotherapy, is quite similar, or to docetaxel or irinotecan is quite similar. So you have the opportunity to treat patients with a monoclonal antibody as a single therapy, sparing all the side effects of a chemotherapy with a similar impact in terms of overall survival. This is the RAINBOW trial. So ramucirumab plus paclitaxel, weekly paclitaxel versus weekly paclitaxel and placebo for patients with metastatic or local advanced unresectable gastric or gastroesophageal junction adenocarcinoma, progressed to a first-line treatment with a cisplatinum-containing regimen. There were 3 stratification factors: geographic region, measurable versus non-measurable disease, and time to progression after the first dose of first-line therapy. These were the key inclusion criteria that are quite similar. Of course, no prior treatment with an anti-angiogenic agent was a major exclusion criteria. The primary endpoint was overall survival. It's not so common in the metastatic setting to define as primary endpoint overall survival. In many trials, the primary endpoint for all solid tumors, progression-free survival is the primary. So it's very ambitious, this trial. These were the patients included with the race, the region, age, gender, and also the weight loss that was a parameter. The time to progression to the first line was less than 6 months for 75% of patients. So this is a poor selection of patients in order to obtain positive data. And measurable disease was present just at least in 80% of patients. And these are the data. So the RAINBOW trial reached the primary endpoint that was overall survival, with a clear difference between ramucirumab versus the placebo arm. It's not so common to find, despite its very, very low the difference, but in metastatic gastric cancer, to have a difference between an arm and another in a second-line setting in patients progressing to a cisplatinum-containing regimen, it's not so common. And in all the subpopulation, in all the stratification factors, we have quite a benefit from the anti-angiogenic agent. And these are the difference in terms of progression-free survival that are quite better, of course. In terms of side effects, they are very common respect to other anti-angiogenic agents. When you combine weekly paclitaxel, a monoclonal antibody that is anti-angiogenic, you have much more neutropenia. Also in the first trial in breast cancer, comparing weekly paclitaxel plus bevacizumab versus weekly paclitaxel alone, there was higher incidence of neutropenia. And also the incidence of hypertension was quite similar to the other arms. There is no increase of death due to adverse event in the arm of ramucirumab. So finally, the safety profile is quite similar to other anti-angiogenic agents in other settings. So the RAINBOW trial met the primary endpoint, and the drug has been just approved as monotherapy for the treatment of gastric or gastroesophageal junction cancer in second-line setting. And this trial finally demonstrated for the first time that an anti-angiogenic therapy can be effective in patients with gastric cancer. What's about other potential treatment, of course? These are all the drugs under development, actually, in my institution and in other institutions in which also patients with gastric cancer are eligible. So we have several types of PI3 kinase inhibitors, from the pan-PI3 kinase inhibitors to the alpha-selective, beta-selective inhibitors, AKT inhibitors, then we have mTOR inhibitors, fibroblast growth factor receptor inhibitors that overall are also anti-angiogenic agents, PARP inhibitors also in gastric cancer if you have a BRCA1 or BRCA2 mutation, BRAF inhibition, despite it's quite unusual, the BRAF mutation in gastric cancer, and then MEK, c-MET, and epidermal growth factor receptor inhibitors. We know the data from HER2 positive disease with trastuzumab in gastric cancer, despite a trial in the adjuvant setting has been never completed, and I don't know if it will be never done, because Herceptin in a few months will be a biosimilar. Finally, will lose the license as drug patented by Roche. So the general idea is also to test the opportunity to do a trial in the adjuvant setting with trastuzumab for HER2 positive gastric cancer. We know very well that if you have an HER2 mutation, you can use other drugs like neratinib specifically. Also, we have specific drug anti-insulin growth factor receptor 1, gamma secretase inhibitors, despite Notch is not a so common alteration in gastric cancer, and Wnt. And then finally, I would like to mention all the national or institutional molecular screening programs that are ongoing all over the world in order to identify molecular alteration and to candidate the patient to programs of phase 1 trials. So we have some in the Massachusetts General Hospital. What is very interesting is that in this program, they have specifically somatic mutation for 14 oncogenes, so there is no platform on a wide genome analysis. In the Dana-Farber Cancer Institute, there is an investment of $43 million to test 470 mutations in 41 genes. In the MD Anderson Cancer Center, there is the Sequenom platform, and there is this program that is the T9 program, a panel of 40 or more genes on a Sanger platform in order to screen something like 10,000 tumors. Then we have Nashville, in which they have finally a program for 40 mutation on non-small cell lung cancer and melanoma, and then a PI3 kinase panel for breast cancer. Michigan University and Princess Margaret Hospital, this is in Canada. So any institution is creating something like an OncoCard in which they study all druggable molecular alterations. So in this case, a whole exome sequencing in Michigan, and an OncoCard with 277 mutations for 21 genes in the Princess Margaret Hospital. And then we have also a program in Europe, 3 institutions. The first one is Cancer Research UK, in which there is breast, melanoma, prostate, ovarian, colorectal cancer, non-small cell lung cancer. This is a stratified medicine program to include 9,000 patients in Center Cancer Centers across UK. And this is finally a reality. In the Institute Gustave Roussy, there is a Sanger platform, and there is also a liquid biopsy program. There are 2 trials active. One is the Moscato trial for lung cancer, and the second one is the Sapphire-02, that is respect to the Sapphire-01 that was a prospective trial, not randomized. This is a randomized trial, so patients with breast cancer are sequenced. You define a molecular alteration, and you randomize the targeted therapy versus observation on patients on stable disease. And finally, the Dutch program. And to conclude, I would like to tell you that in our institution, we are trying to create an OncoCard. The general idea is to create a card in which we insert just the druggable pathways, so not mutation for which there is no available drug. And the difference with the other cards is that we will include also amplification, specific amplification that can be druggable or actionable, let's say. And this is my last slide, and I will take questions if you have. Thank you very much.
You didn't mention anything about immunotherapy. Now, I'm bringing that up because the issue about drivers and passengers, in my mind now, is really beginning to change. Because you can have mutations that, in fact, are so-called passengers. They're not drivers. And yet, some of those mutations may give rise to mutant proteins that can be targeted by, for example, immune checkpoint control antibodies and so forth. So how do you feel that that's impacting precision medicine?
So I have my personal perspective on immunotherapy. We have several trials here in my institution on immunocheckpoint inhibitors, specifically one with the agent that is the MedImmune agent, that is an anti-PD-1, and another one with pembrolizumab in triple-negative breast cancer. I really believe that when we speak about immunocheckpoint inhibitors, we have two different points to be considered. The first one is the mutational burden of the cancer. So the idea is that much more genetic instability you have, much more immunogenic is the tumor.
Yeah.
But I have another perspective. I really believe that it's not just a matter of mutational instability, but it's a matter of neoepitopes that are created on the tumors. So in order to have an immune response, you don't need just a high mutational burden, but you need the tumor to express neoantigens that are immunogens. And the recently data published on the New England from the group, from the United States group, specifically on melanoma, clearly demonstrated this. So you have patients responding to ipilimumab, in which you have a response just if you have the expression of neoepitopes that finally are viral epitopes. This is very strange, that induce an immunological response against the tumor. So in my opinion, the idea to include in a phase 1 trial just patients that have PD-L1 expression, without considering that PD-L1 expression is dynamic, is heterogeneous, can be expressed in the tumor, in the T cells, and in the stroma, this will deny the access to the immunocheckpoint inhibitors to patients that can benefit. And if you consider all the trials in lung cancer, in melanoma, and in solid tumors, we have patients without PD-L1 expression that respond despite the negativity. So this is my personal perspective on this point.
What's happening in the area then of gastric, carcinoma? Are there trials now? Is there any indication, any hint?
This is a good point.
Yeah.
In our trial, the MedImmune trial, there is a cohort with 30 patients with gastric and gastroesophageal junction. The expression of PD-L1 in the tumor was around 5%, and the response rate was around 10%. That is quite good if you consider that these are patients refractory. The only patient that we included, I have to tell you, it was a patient with gastric cancer, with a brain mets, and he has a response for 4 months, and then he progressed with a new brain lesion. So my experience is on one gastric cancer patient, PD-L1 positive, that had a response to this MedImmune anti-PD-L1.
Yeah, one reason I was asking is that, at some recent meetings that I've been at, there's some hints, I would say, rather encouraging, it's still early days, about combining, bevacizumab, by way of example, with nivolumab.
I have to tell you one thing.
Yeah.
There is the Triatec grant from Merck, and we applied, and we proposed a trial for patients with lymphangitic breast cancer with pembrolizumab plus low-dose cyclophosphamide, metronomic chemotherapy, because the hypothesis is that low-dose cyclophosphamide can downregulate Tregs.
Right.
and upregulate the CD8 cells. And the grant was approved by Merck. They will give us substantial financial support, and they will give us pembrolizumab. So we will start with this trial, combining low-dose chemotherapy and pembro.
Would that be a single, I know that there are groups in the States, again, like Steve Hody, that are doing something similar, but using a single administration, just one shot of the cyclophosphamide. Is this going to be continuous?
We do metronomic.
You're going to do true metronomic.
They enjoyed the idea to do something like this.
Okay, good.
We had an earlier crack at this discussion, but I'd like your view about it. This is the notion of combination of anti-angiogenesis, best in the field for a particular organ with chemotherapy. And I think you suggested sequential versus together and so how are we going to solve this issue? Because I have the feeling there's something there, but we just can't get at it without empirical, endless combination trials and so on. I mean, it's just a little bit mindless. With all the science
and anti-angiogenic. So we did one trial. The trial was a prospective randomized trial comparing sequential bevacizumab followed by chemotherapy versus concurrent. And it has been just published a few weeks ago, and the paper was rejected in sequence by JCO, JNCI, Annals of Oncology. Despite I am a member of the editorial board, so I listened about the discussion also.
Don't worry.
And I took no position, because they told me, but why you are designing a trial with a sequential approach? It's unethical to give bevacizumab alone to a patient with a metastatic breast cancer when you know that concurrent administration is better. So in our experience, I can tell you that giving to these patients the sequential approach, we observed some response, first of all, and we performed biopsy at baseline, at response, at the progression, and we found a gene expression array in which you have upregulation of genes involved in the matrix remodeling. It was very clear that were upregulated before BEVA administration and downregulated following BEVA administration in case of response. If anti-angiogenic agents may normalize or not the vasculature in order to better deliver the drug, it has not been clearly demonstrated this. Our trial was the only attempt to demonstrate this, but finally it has been published on the breast, 2 of impact factors, so nobody understood this attempt finally. And in my personal opinion, I believe there is a role still for anti-angiogenic agents. We have to deliver them in a concurrent fashion because they are approved to be delivered in a concurrent administration. But in some cases, drugs like ramucirumab that can be effective also alone with a single approach, this is a clear demonstration that an anti-angiogenic agent may be combined rather than with a standard chemotherapy, with a metronomic chemotherapy. Let's say, I am quite sure that an anti-angiogenic agent will be more effective with a low-dose chemotherapy rather than a standard dose chemotherapy. This is my opinion.
One thing about the sequencing, sorry, Gordon, if I could just, it seems to me that one difficulty about this is that if you're using an antibody, which has a long half-life, weeks, I think the idea that you're going to sequence chemotherapy with the antibody is going to be, by definition, it's not like you give one and then you give the other. If one is an antibody, it's going to be around virtually all the time to a certain extent. So I think with TKIs you could sequence, but that have a very, very short half-life. But I don't see how you could do this in a practical way, with antibodies. And I think that's another reason why concurrent administration just intuitively makes sense.
I was going to just turn back to you in terms of your role maybe in monitoring that sort of trial.
Absolutely. I mean, I think from a standard view of monitoring, you're going to have to use, of course, RECIST and there's nothing else. But I think there is a opportunity to look at the
DCE MRI,
diffusion MRI, and some of the more advanced techniques that are coming out there to give you concurrent mechanistic information, which can hopefully start answering some of these questions about has there been normalization in the vasculature? has there been a change in the cellular structure? I mean, some of the more advanced diffusion imaging can actually tell you about cell size, as well as cell density, as well as extravascular space, as well as vascular volume. And so that sort of power to get the mechanistic non-biopsy confirmation of changes ongoing during the treatment in particular sequential or concurrent trial, I think quite, adds power to the overall trial design.
I completely agree with you, despite, I remember the seminal paper on bevacizumab in rectal cancer, in which they perform a DCE MRI in order to study the pharmacodynamic effects. So if you want to look at action of beva, at least, you need to perform MRI on day 12, because the maximum pharmacodynamic effect was on day 12, if you remember that trials. So I am sure that anti-angiogenic agents work. Now we have a trial with lucitanib. Lucitanib is finally a fibroblast growth factor receptor inhibitors, but it targets also vascular endothelial growth factor receptors. And we have something like 15 to 20% of response rate in patients refractory to chemotherapy. And the activity is, in all patients, angiogenesis mediated, because you have the tumor lesion with a cavitation in the middle. It's typical response of sunitinib or similar drugs. So, bevacizumab failed in some diseases. I don't know if failed, because in breast cancer is a very active agent, but despite this, the comparative effectiveness research from FDA and the NICE decided that it's not a good drug in breast. But there is an increase in progression-free survival, like ramucirumab. There is a response rate that is higher respect to paclitaxel alone, but the regulatory agencies decided that it's not a good drug.
Well, I think before Adriana was going to ask something, I think one of the mysteries to me about the FDA decision, it's not so much the FDA decision, but maybe Genentech and Roche. As a biologist, if I'm asked to repeat an experiment, you repeat the experiment. And the experiment here that was asked by the FDA to be repeated was weekly paclitaxel with bevacizumab. And for whatever reason that I'll never understand, what was done was to then do the Avaro trial with once every 3 week, docetaxel, or trials ribbon 1, ribbon 2 with capecitabine or with an anthracycline. The weekly paclitaxel trial was never repeated. I don't understand why that was. I think there could have been variations of that trial. It wasn't placebo-controlled. There could have been a maintenance bev after the upfront chemo plus paclitaxel. So I still think that trial, a very interesting trial, E2100, and it's unfortunate that a decision was made for whatever reasons, marketing or otherwise, that actual precise protocol was not repeated when I think it should have been. Anyway, that's another story. Adriana.
But in the RAINBOW trial, they did. It's very well tolerated with paclitaxel.
Weekly paclitaxel.
Yes.
Yeah.
Giuseppe, I wanted to go back to the precision medicine, and I always being a microenvironment person, I always have doubts on the fact that, particularly in solid tumor, every division you have acquired mutations, so you have this issue of very popular of tumor heterogeneity. It's very difficult to distinguish when you do some sequencing, if all the parts of the tumors have those particular mutations. For instance, we had recently, in the cases discussion, breast tumor that had, very difficult parts, different. One HER2 positive, the other one triple negative, and it was in the same breast. So in this context, how do you work with the precision medicine?
So we have the bioinformaticians that should advise us if this is a driver or not driver mutation. So there is the role. I am member of the steering committee of the Aurora, and so every week we have this meeting with Peter Campbell, with people working in Cambridge. And finally, they gave to us a list of mutation in that specific patient. What we have to do is to understand if is there any drug available for that specific genetic alteration, but they don't take into account the tumor heterogeneity. We have just one-shot photograph of the liver met or of the lung met or the brain met, but we don't have nothing about the opportunity to find other mutations in other site of disease. What I can tell you is that in almost all patients, you have so-called private mutations. So in some cases, you have a PI3 kinase mutation in 70% of the patients, but there will be one patient with a BRAF mutation for which you should ask an off-label indication of emurafenib. But will you obtain this in your country? Because we increase the knowledge, but finally, in the real life, you have to find a drug for the patient. And in most of the cases, you don't have a clinical trial, or you don't have an access to that specific drug because the regulatory agencies registered the drug for another indication. So it's not so easy to do precision medicine initiatives with the actual regulatory board that provide the drug with a specific indication within one disease. I believe drugs should be approved according to pathways and not to disease. But this is a political issue, it's not a scientific issue.
We haven't heard anything, unless I was out of the room, on ovary.
Ovary cancer?
Ovary, yeah.
I would like to mention just one study on ovary cancer. It was the study with cediranib plus olaparib for ovarian cancer refractory to cisplatin, published on The Lancet. This is a seminal trial because it's the first time that you use a regimen without chemotherapy in ovarian cancer that are not BRCA mutated. And it was a combination of an anti-angiogenic agent, already tested in lung cancer by Pamela Goodwin, plus a PARP inhibitor. And the trial was a positive one. It works. So giving a small molecule like a PARP inhibitor with another small molecule that is an anti-angiogenic agent can induce response in almost 20% of the patients. Which is the mechanism of action? I really don't know. What I can tell you is that George Kukos publishes some data on Nature, for him, he's an immunologist, demonstrated that an anti-angiogenic agent like bevacizumab can downregulate the T regulators and can enhance the tumor infiltration lymphocytes within the tumor. So another mechanism of action of an anti-angiogenic agent is enhancing the immune response. I do not suggest to use bevacizumab with an anti-PD-1 because colitis or gastrointestinal complication can appear in 10 to 15% of patients. So if you have also an anti-angiogenic agent, the risk of bowel perforation is a reality in these patients. And so following colectomy can be proposed. So, but anyway, this is another example of interaction between drugs.
I was just trying to get you to answer Bob Kerbel's question about immunotherapeutics, which you've just done. So, absolutely, I think it's.
No, in the MediWound trial, there was a cohort for high-grade ovarian cancers, again PD-L1 positive, so this is the limitation of this trial. We did not include patients, but the rate of response was less than 5%, despite high mutational rate of high-grade ovarian cancer. But immunotherapy for the future, I believe, will have a lot of opportunities in many tumors. Sure. They approved on a basis of a phase 1 trial, nivolumab in non-small cell lung cancer, second line. So it's quite impressive.